E aí, pessoal, boa tarde, sejam todos bem-vindos. Mais uma transmissão da Liga Acadêmica de Histologia e Biologia Celular, da UFG. Meu nome é Pedro, eu sou o coordenador da Liga Acadêmica. A Liga Acadêmica de Histologia e Biologia Celular, para quem não conhece, ela é uma iniciativa que surgiu dos alunos de graduação da Universidade Federal de Goiás. E a gente ele é um projeto de extensão que a gente tem como objetivo principal fazer divulgação científica, principalmente de assuntos relacionados com histologia e biologia celular. Então, periodicamente a gente promove alguns eventos, alguns seminários. Atualmente, os nossos eventos têm sido todos online, devido à pandemia. Mas isso, na verdade, tem aberto uma oportunidade bastante interessante para a gente, que é exatamente a gente conseguir convidar pessoas de fora para poder falar com a gente, conversar e fazer a sua palestra. Uma das atividades que a gente faz na Liga Acadêmica também, entre os membros da Liga Acadêmica, é a discussão interna de alguns artigos que a gente acha interessante da área de ciências biológicas, biomedicina, histologia, biologia celular. Recentemente, a gente discutiu um trabalho que todo mundo achou bastante interessante, que dizia sobre o uso de inteligência artificial na microscopia, como a inteligência artificial pode ser utilizada na microscopia, como ferramenta para a microscopia. Como foi um assunto que despertou bastante interesse entre os membros da Liga, a gente pensou, por que não convidar o autor principal do trabalho para dar uma palestra para a gente? E fizemos o convite e ele aceitou, gentilmente aceitou. Então, eu vou convidar ele para dividir a tela comigo daqui a pouco. O nosso convidado é o Lucas von Chamier, ele é um estudante de PhD, né, em Londres, né, na University College London. Então, ele na verdade, ele é alemão, mas mora em Londres há alguns anos já. Então, nossa palestra hoje vai ser em inglês, vai ser a primeira transmissão em inglês da nossa liga. Eu vou convidar o Lucas, então, para poder compartilhar a tela aqui com a gente, começar uma transmissão, e a partir daí eu vou começar a falar inglês. Lamento se meu inglês não for muito bom, peço desculpas já, mas eu vou me esforçar bastante para não cometer nenhum erro. Tudo bem? Então, vamos convidar o Lucas.
Is that my cue?
Lucas, you're online now.
Perfect. Thank you.
So I was making a brief introduction about how we know your research. You were it was a discussion about the paper you wrote about artificial intelligence in microscopy. So at the beginning, I wish to thank you very much for accepting our invitation. It's very important for us. It's very excellent opportunity to. It's the first time we have an international speaker in our academic league, and you accepted our invitation. You're very gently accepting our invitation. Lucas, let me make a brief introduction about your research, and you can correct me if I say any mistake, okay?
Sure, go ahead.
Lucas, his research is focused on developing deep learning methods for microscopy, and he tries to adapt the existing deep learning technologies for application in bioimaging. And he has a special focus on creating simple workflows and quality control. curious fact, he's also like bird watching, and he's very interested in conservation, biodiversity. What I believe it's very interesting, because all these areas in my mind, they are all connected. Lucas, it's okay?
Yes, that was perfect. Thank you.
No, I said no mistakes. Lucas, so thank you very much for accepting. If you want to say something, or if you want to begin already the presentation.
Okay. Can you see the slide, the first slide?
No, because I didn't share it already. Okay, now it's on mine.
Perfect. Wow, thank you so much for this introduction. Yes, my name is Lucas von Chamier. I'm a PhD student at the Enriquez Lab in London, and Pedro gave a wonderful introduction, and I'm really honored to be the first international speaker in your seminar series, and I hope you get many more speakers to talk to you in the future as well. And so, without further ado, I would like to introduce my research to you, and I will try to speak very slowly, because I understand there might be a little bit of a language barrier, but I hope you can follow the slides anyway. And if there's any questions, I hope that later I can answer some of them. So my research has been about using AI, or a subset of AI methods called deep learning, for microscopy. And I've developed with some collaborators this project called zero-cost deep learning for microscopy. So to give a brief introduction why we're interested in deep learning, we need to talk about the challenges we have for imaging first, and many of you might be familiar with some of these challenges. One specific challenge in microscopy, specifically for live imaging, is phototoxicity and bleaching. So you can image cells in a very live cell-friendly manner at low intensities of light. We're talking about milliwatts of energy. So this cell is very happy, and it can be imaged for many minutes and nothing really happens. But for super resolution methods, you often need very high-powered lasers, and that ensures that you get high signal. But what happens then is your cells can die. And this is due to something called phototoxicity. As you increase the intensity of light being focused on your cell, the more damage you can do inside cells, and the more you trigger phototoxicity responses in the cell, and your cell dies, which is bad. In general, this basically means that your super resolution technique, which might image your cell, is a little bit more like one of these war machines from War of the Worlds, and this is what you're doing to your cell, which is not desirable. Another problem is that you might bleach your cell. Your fluorescence might decrease over time, and you just lose signal, and you can't see anything anymore. This is a failed experiment of mine, where my cell basically becomes invisible after just a couple of minutes. So what this means is that we're basically faced with this kind of dilemma, where we can either have very live cell-friendly methods, where your cell stays alive for a long time, like here, but often we use very low energies for it, and we will have a lot of noise, and our images might not have super resolution. Techniques that use this kind of range are, for example, bright field methods, which is just normal white light. Super resolution often sits up here in the kilowatt range, which basically is lethal to many cells over long periods of time. And most fluorescence methods sit somewhere in the middle, where the amount of damage you're doing to the cell isn't quite clear always. Similarly, for temporal resolution, so how many images you take per time, follows a similar rule. Basically, the higher your temporal resolution, the more toxic for the cell, because you're adding more noise to the environment. So this is one big challenge we're trying to solve in microscopy today, which deep learning, as I will show you, can help us with. Another big challenge is throughput. So if you have an image, for example, of many nuclei, and you want to analyze these nuclei, this might take a long time for a human to do. And this gets even worse if you have to analyze many, many, many pictures of nuclei. This would take weeks for a human to do. And that's why scientists are interested to find methods to automate this process of analyzing this data. So how can deep learning help us? Why are we interested in deep learning? Well, deep learning is really good at analyzing images compared to other algorithms. And one example for this is this kind of movie of what a self-driving car can see in an environment. You might think of, Tesla or some other companies that are making these cars, and they're incredibly good at identifying objects in very complex image data. So this is one reason why scientists in microscopy are also interested in using these powerful algorithms to analyze their data. And indeed, in the last few years, hundreds of papers have come out that are using deep learning, and this has caused a huge revolution in microscopy. So what is deep learning and why is it so good for cell imaging? So if we compare deep learning with like a classic algorithm, let's say for denoising, this is a very common task where we try to remove noise from an image. We use an algorithm that can do something with this image, and we have programmed this algorithm, and we get a denoised image out. This is the classic case. In deep learning, we use something called neural networks. And often the architecture of a neural network looks something like this, with different blocks or layers that perform sort of different tasks with increasing complexity. And what we need to do to use this is we give it a training input and a training target, and both are fed into this neural network. So note the arrows. And as we add more and more images of this type of data, the neural network will learn how to translate the left side into the right side, into a denoised target. This usually happens over a couple of periods of time, which we call epochs, where the neural network will see this data from both sides over and over again. And as you kind of go into the network, this is what it looks like. Each layer in this neural network is usually something called a convolutional layer that will perform convolutions on the input image. A convolution is basically just a mathematical operation that changes the data or the representation of the data and extracts certain patterns. And that can, for example, look like this. And you can do more and more of these convolutions and steps called pooling layers, which will simplify the data and reduce the amount of pixels until you get something very, very abstract. But it's simple. It's a simple abstract form of the input image. And your neural network can learn these abstract features and basically translate them into this type of target image. And as you go through this process over and over again, your algorithm or your deep neural network starts getting better. In the beginning, it might do pretty badly at doing this translation, but over time, it will start getting better until it starts really being very good at this task. And this is what deep learning kind of looks like in principle. So you get loads of training data until your algorithm understands the task it needs to perform. However, this is computationally very expensive, not just computationally, also in terms of money. And that's because a lot of these processes use something called GPUs, a graphical processing unit. And deep learning needs these very powerful computational processing units. because this is a difficult task and takes a long time. Another problem is that when you try to use deep learning, you have to go through these, often you have to go through these GitHub pages, and then you have to look at a lot of this code until you can use the algorithm. And that's quite annoying, especially for biologists who might not have the time or the expertise to go through all of this code. So it's also challenging from a user perspective. This is why a couple of different groups have started to make more accessible deep learning projects for microscopy. One of them is CSB Deep. We also have Deep ImageJ, which is basically where you can use deep neural networks that are trained in ImageJ, which is very commonly used in biology. And there's also something called Elastic, which also has like really accessible tools for biologists. Another problem is that it's not always easy to train this kind of deep neural networks on your own data. There's often no clear explanation on where to start and where to finish. And there's often no quality control, or it's difficult to know if you can trust your algorithm. And the problem in that is kind of depicted here. If you have an algorithm that can sort of recognize patterns in a very pixelated image and produce something that looks really good, how do you know that what's on the right-hand side is really what is underlying here and not something completely different? So we have a trust problem in biology for deep learning tasks, because how do you know that your algorithm isn't putting out something that is completely wrong? This is why quality control is very important, but it's not always available. And as I said, money is another issue. This is where my project comes in. We tried to make a platform that can address some of these challenges. And specifically, we wanted to build a platform that reduces the cost for the user, makes deep learning more user-friendly, and makes the results that we get more trustworthy. So the first step that we did was find a good backend or a good platform to do this. And our idea was to do this in Google Colab, which you can access through a Google browser. This is for free, and you get access to GPU, which you need to do deep learning. This is really important because you need to train a network on your own data to get the best performance. So GPU is really essential, and Colab gives you this for free. Next, we decided we needed a nice interface. And here's a short sort of introduction video of how the interface looks and how you can access this tool. So we have a GitHub page, and on our GitHub page, you can basically get access to the Google Colab notebooks that are just in a web browser, and it looks kind of like this. You can make a copy of the notebook, and then you can start training it, giving it your own name, etc., etc. It's all code-free, and we also have some training data sets to basically show you how to use the notebook. Our data sets are published on a page called Zenodo, so you can have a look what our data sets look like, or you can just start with your own data set. It doesn't really matter, because it should be just as easy to use on your own data as on our data. What you then can do is you can upload your data to your Google Drive if you have a Google Drive. If you don't, you should make one, because that's sort of one of the main, that's at the moment the main way to use the notebooks. Then you can upload your data. I can skip this part a little bit, and then we can start using the notebook. And that's pretty easy to do, because all you really need to do is play the cells and connect it to your Google Drive, which is done here. You have to go through a few steps to connect it to your Google Drive, which happens here. And then once you're connected, you can basically get access to all your files, including your training data sets, in your Google Drive here. And then you can install the kind of method that you want to use, in this case, Stardust, which I will show later. which can do segmentation. Sorry, I skipped accidentally. And then you can put in your training data sets, which is basically just a path in your Google Drive. And you can tell the model where to save it, where to save the model. You can play these cells. It will input the training parameters that you want. Then you can create your model and your data set. And finally, you can then train your model. And all of this is just by playing the cell. You don't have to code anything yourself. And we also have quality control implemented, which I talked about earlier, which can help you trust the output of your model. For that, you can input a data set for testing. And you can basically see how well your model performs on a new data set. We can also check out how well the model learns during training. This is kind of basically shown here. And here is where you can input your test data set, where you can test on new data how well your model performs. In this case, this is a segmentation algorithm. So we want to see that it detects all the cells and can basically draw a mask around all the cells' nuclei. And what you can then see here is how well this overlaps. And then finally, you can also use the model on completely new data, data for which you don't have a mask yet. And that's kind of the point of the whole deep learning exercise, where you can basically use unknown data and use your model to predict something. And that works in the same way. You put in the folder where your files are, you say which model you want to use, basically, which is this little button. And then you can say which model you want to use if you don't want to use the one you've just trained. Then you press play, and then it will analyze all the images that are in these folders. And that's it. That's basically how our platform works. Oops. And basically, just to wrap this up, the workflow is really simple. So you have to just prepare a data set, prepare your network for training, train your network, evaluate using quality control, and then use your network. And the quality control for images is also implemented. This I might skip over this a little bit because maybe this is not too relevant right now or goes into too much detail. But basically, what you can do is when you have a prediction, as I showed earlier, from your model, you can have a look how close this prediction is to your target, which is the ground truth. You want these to be as similar as possible. So how can you test this? Well, we have implemented a couple of methods that can basically compare how close these images are to each other. One such method is basically just pixel differences, which measures how close are these pixels to these pixels. And if the difference is very large, it will be bright. And if the difference is low, it will be very dark. So here we have like a very low pixel difference. This is the mean squared error, by the way. And we also have a method called structural similarity, which measures the structural similarity. So how similar are the structures on the image between these two? And here a high value is good. And our algorithms can basically, we can show that in this case, our prediction is really good. We can also do this between the source and the target, but maybe that's not that important right now. Basically, we have these quality controls for image-to-image tasks implemented. And then once you have this kind of pipeline, you can test how well your model performs with your quality control. And then you can go back and improve your model and test it again, and improve your model and test it again, and so on and so on. And once you're happy with your model and you trust it, you can use it for step 5. That's kind of how we intended our system to be used. Okay, so let's have a look at some of the tasks we now have implemented. So as I said before, we have some segmentation, denoising, and also some really nice new tasks that use artificial labeling. Segmentation is basically just exploring how like foreground objects from background objects. And a very simple implementation is something called UNIT, which can, for example, segment these membranes of I think this is a Drosophila hippocampus, or sorry, Drosophila brain or something like that, but in electron microscopy. Stardust can do nuclear segmentation, which I showed you earlier, which can take ages for a human to do. And Stardust is specifically good at identifying nuclei that are almost touching, or are touching or overlapping, which many non-deep learning algorithms really, really struggle with. So Stardust is one of the best nuclear segmentation algorithms and is really, really efficient and works extremely well. You can even analyze with our platform moving cells and cells and movies, which is usually almost impossible to do by hand. For image denoising, we have one algorithm called CARE implemented, which uses basically the system I showed in the beginning. And you can see here how the algorithm can kind of restore the object that is really noisy in this image. We can also do this in movies in the same way as before. Another algorithm is noise to void. The advantage of noise to void is that we do not need to use ground truth data. It can basically learn the noise just from the ground, from this noisy image, and then remove that noise. So this is a really exotic, amazing task. And we've also successfully implemented that in our notebooks. Again, here's an example of how this can look in a moving picture, in a time lapse. So we've also implemented this in 3D. So you can also use this on 3D data, which is pretty awesome. I'm not sure how well you can see this here, but on the right side, you can see what the denoised version of the left-hand side looks like if you use deep learning, which is obviously amazing. So as a summary, here are like the algorithms that we have that I've shown you. Another one that I didn't show now is YOLO v2, which can do classification. So it can tell you what type of classes or cells you have in your image. So for example, elongated and rounded. These are some of the denoising algorithms that I showed. We also have one that can do super resolution reconstruction, which is basically resolving images beyond the diffraction limit of light. To explain this would take a little bit longer today, but you can have a look at it yourself on our GitHub page. And finally, we have these image-to-image translation algorithms, one of which is FNet, which I think I showed on my first slide. And we also have these really cool algorithms that can kind of do any image to any image. And one example we chose is, for example, to predict nuclei just from membrane labels, or I think maybe this is actin cortex. So you give the algorithm an actin cortex and it can predict where the nuclei are, which can, for example, help you when you want to track your cells. And we have another algorithm that can do something similar, but here we've used an algorithm called CycleGAN which we trained to restore a sort of a non-super resolution input into a super resolved output by using structural illumination microscopy. So all of this together is the zero-cost deep learning for microscopy platform. So what have we done? We've integrated many different deep learning algorithms. We've reduced the cost because we've implemented it on a free browser platform. We've made these methods more user-friendly by reducing the amount of code that you need to use these methods. It's actually very, very very easy to use. And we've implemented some quality controls so we can trust the results more that come out of these algorithms. These are the algorithms that I showed you that are implemented now, but our platform is always growing. And if you have an idea or you want to contribute, you're welcome to get in touch with us. There are some recent additions that we've been working on that you can also try. And we're also working on more algorithms that can do different tasks that we don't have implemented yet. These are the names of these algorithms. So here are some examples how people have used our algorithms. So one of our collaborators or somebody who's been working with us is Christoph Spahn. He's been using our YOLO notebook to classify bacterial colonies in his images. So by using our algorithm, he was able to basically figure out how many colonies are in his images, when they occur in the time lapse, so how fast they grow. And he's also done this to check out how they respond to drugs and what phenotypes come up when he uses different, for example, antibiotics. And so you can see we're kind of, we are overlapping with what we wanted to do in the beginning, which is basically making it possible for biologists to get access to some of these cool deep learning methods. And other users, for example, on Twitter, to show a few screenshots, have been using our tool. And I think this shows that like it really can help users to use deep learning. And that's it. I would like to just mention a few people by name. Who hasn't made it on this list is Isabella, who got in touch with me on Twitter, which I think is an awesome way to get in touch with scientists. And also Pedro, who's been very kind for inviting me. On this list are the people that have been helping me on the project. So my supervisor, Ricardo, and also my main collaborator, Roman, who's a postdoc in my lab. We have also got collaborators from Turku, which is in Finland, especially Guillaume Jacquemet, who is a cell migration expert. And then here are a couple of other people that have been helping us as beta testers and just for general advice. So, for example, Christoph Spahn and also the CSBD group from Dresden. And this is my funding body that funds my research and the name of my institute. And that's it. I hope you enjoyed it. I hope that it wasn't too fast. And please use the tool. I hope that like this is something that can be useful to all of you. And if you have any problems, get in touch with me or anybody else involved. Thank you very much for listening. Yeah.
Lucas, it was very nice. Very nice. Congratulations.
Thank you.
The comments of the audience were very interesting. Fantastic and amazing were some of the words they were saying about your presentation. Congratulations.
Okay, cool. Do you want me to answer any questions or something? I'm happy to.
Yes, there are some questions. If you don't,
I don't mind at all.
Well, I try to put here at the screen. Which program language do you use in this project, and how many images do you have in your DB?
Okay, so program language. We're using, basically all of this is based on Python. Actually, most deep learning tools today are based on Python. There are a couple of different packages that are kind of competing at the moment for, I guess, supremacy. There's TensorFlow, which is developed by Google. And actually, most of the methods that we've been using use TensorFlow as a deep learning backend. There's also a couple of tools that use PyTorch, which is the competing deep learning language made by Facebook. So it's basically Google versus Facebook. And at the moment, most tools are made for microscopy are made with the Google backend. That's my experience. But Python is the underlying thing that all of these languages use. So Python is the one. And how many images do you have in your DB? I suppose, I don't know, DB is the Google Drive, I suppose.
I'm not sure.
But basically in the training data sets for our platform, we actually don't need that many images. So like we had surprisingly good results with only 30 images for some of the algorithms. More is usually better. So the more images you have, the better your algorithm will be. But it's useful to try it with fewer images. Like you can basically get started with just a few images. If it doesn't really work, you can always add more, but you can already see quite nice results with, less than 100. Obviously, it would be awesome if you can have 10,000 images, but that can be very difficult, right? So maybe 10, 20, 30, 40 images you should start with, Some algorithms like noise to void, for example, You only need one image to train it. So it depends on the method as well.
Well, nice. I was figuring about hundreds or thousands of images.
No, actually, I should have mentioned this probably. Obviously, so one of the limitations of the project is that the Google Drive obviously has limited space. So I think for like a free account, it's 15 GB. So you have to sort of think what kind of data you can put into that amount. But for some segmentation problems, for example, like sort of drawing masks around cells, for example, usually people don't have such huge data sets. So it's usually a couple of GB. And then you can really use these notebooks quite efficiently. But the limitation is the amount of space. So for really big deep learning projects, this might not be the right thing, but it's a good way to get started. Really good way to get started, I think.
Nice. There's another question from Giovanna. Giovanna asked how difficult it was for you to learn how to work with artificial intelligence within biology. I mean, by learning the coding and upgrading the codes, comparing results and stuff.
Yeah, I have to say it was quite difficult. I'm not a computer scientist. I'm a biologist. I have a little bit of a physics background from my bachelor's, but I didn't do any coding before I started this project, and that's part of the reason why I made this project. It's because I found it really difficult to use AI, and I wanted to make something that's easier to use. And I hope I made a tool that like somebody could understand, like that is like me for like 3 years ago. Like I wish that this project had existed when I started doing this, because it would have made it so much easier to get started. I can recommend, like if you don't know much about deep learning or AI in biology, I can recommend my project, because I think it gives you we give a lot of sort of tips and I like sort of feedback on how you can use AI. And you can look at our code as well. The code is always inside the notebook. Just double click on the cells and you can see the code. So if you want to know how these models work, it's not easy, but if you know a little bit of coding, you can get into it relatively quickly.
Giovanna, her comment continues. It looks almost impossible to work with AI without knowing a thing or two about both the universes, biology and coding.
Yeah, I mean, I would say as a biologist today, you should probably learn how to code. And you don't necessarily need to do deep learning, right? But it's already useful if you can automate some of the tasks that you're doing.
Yes.
If it's just whatever, like repeating a process over and over, like repeating the same process on different files, for example. Python is really good at it, but ImageJ, if you're using that, also the macro language there, very useful. But for AI, Python. You have to learn Python if you want to use AI. But you don't have to learn anything if you want to use our platform. So that's the whole point. I think it's horrible that you have to be able to learn how to code and understand the code if you want to use deep learning, because deep learning itself is actually very straightforward. It's like you do this, you do that, you have like a training data set, now you want to train. So it should be just as easy as what we did, which is pressing play and letting the code do its thing, and you don't have to worry about the code. So yeah.
And it's very nice because you created a platform for users and not for developers.
Very much, yeah. But developers are also encouraged to, I think like what we're trying to tell developers is, please make your tools in a way that more people can use them. Because as I said, there are like hundreds of deep learning tools, but some of them are so hard to use if you don't know like this kind of project, and it's that defeats the purpose. It should be, the best science is possible if many people can use the tools, I think. So yeah, a little philosophical rant.
No problem. Lucas, there is another question from Domingos Alves. He asked, what would change in your work if you preferred to apply ML instead of TL?
Machine learning instead of deep learning.
Yes.
That's a good question. So to be honest, I'm not that familiar with machine learning itself. I've focused almost entirely on deep learning. However, I know how machine learning itself, roughly works. So machine learning is a more classical sort of approach to, I guess, automating difficult tasks. So you can, for example, have decision trees or random forests or something like that. What would change? I think, for example, random forests and these types of algorithms, they cannot do some of the tasks that deep learning can do at the moment. So, for example, the artificial labeling that I showed, so where you predict a fluorescence signal from a bright field image, or just translate a random image to a different random image, is really something that I think has only been done since deep learning started. So I think if I prefer to apply machine learning, I think I just couldn't do some of the things that I'm doing now. On the other hand, deep learning is very hardcore computational, and if you can find something that can achieve the same results without deep learning, it's always better to do that instead of deep learning, if that makes sense. Because deep learning is very complicated and complex, and it might be wasting resources if you could do it in a simpler way. So deep learning is not always the answer, but it can be sometimes the only answer.
Yeah, makes sense. Nice. We have a person from Chile watching us, Josefa. She said, hello from Chile. Hello, Josefa. Really nice. ¿Cómo estás?
Cool. I'm glad. That's awesome.
And let me see. There is another question, if you don't mind.
No, I don't. I was under time. I actually thought I spoke much longer.
Okay.
So.
Augusto asked, what are the next steps you want to take with your project, and what do you think still needs to be improved?
Loads. Yeah, I mean, there are already a couple of new projects that we're planning to implement. So there are like a couple of algorithms that we want to implement that I already talked about. For example, there's one algorithm that I'm keen on developing, but I first have to kind of finish my thesis. But basically that algorithm can do instance segmentation. For example, you can then see masks on individual objects and also classify those objects. And I think that could be really useful. Then there are like a couple of algorithms that we're interested in, for example, that use more unsupervised approaches, because one of the main challenges is creating a data set, right? Like we've sort of, I guess I wouldn't say solved, but we've addressed some of the main issues of using deep learning. Now the main problem becomes making the data sets. And that actually is probably the most challenging part about deep learning, once you've figured out how to do deep learning, is making your data set. And I think that would be a whole different project. But I think one way to address this would be to have more algorithms that don't need these paired data sets, but algorithms that can kind of use just one data set and do the deep learning task that you want to do just on that data set. I think that's the future of deep learning anyway. And how I think I would like to improve the project itself, like the notebooks or the project itself. There are a couple, I mean, there are plenty of things that could still go wrong in the notebooks. So I think I would like to improve some of the code, some of which I wrote when I was like very new to coding. The code is just a bit ugly in some places, I think, but it works. Yeah. How could I, I think we probably could improve some of the data set stuff. I think that's the main challenge. Like some algorithms only use TIFF files, for example. Other algorithms use JPEGs or PNGs, which is annoying. I think it would be good to make it easier to convert between different data types, for example. That's a useful improvement, I think. Yeah.
It would be very nice.
Yeah. If we have time, we'll do it.
And I hope that you don't stop this project, because I believe it's very important. I'm very interested in trying to use your platform.
Yeah, please go ahead. Go to our GitHub, come to our GitHub page, and if you don't understand something, just write to us or go on our GitHub or Twitter or whatever platform. We're pretty active. We're still improving it. Yeah.
Okay. And there are some comments here from Giovanna, Domingos. They all acknowledging about your presentation.
Oh, thanks.
Domingos also says thank you so much for your answer for his question. And let me see, Augusto also saying thank you for sharing your knowledge.
Can I also say that I don't know, I think it's amazing that you invited me. I'm really happy to give this kind of talk, and you should always reach out to the authors of the papers that you like. And I think many of them would be very excited to speak to you, especially in Brazil, because I think there isn't that much. I think like a lot of research is too focused in Europe and North America. And I think like sometimes that leads to sort of a very closed-minded bubble. And I think it's really important. I'm really happy to sort of share the knowledge with groups that are outside that bubble. So keep reaching out and like grow your network and stuff. I think it's awesome. I'm loving this. Yeah, I think many people would like enjoy talking to you.
Oh, there's a nice question here. If we have some more time. Lucas asked. Lucas is asking if the journals accept researches normally when using this technology, which is still recent and in development.
Okay, so that's a good question. I know that. So I can tell you that like if you make a deep learning method for something that hasn't been done before, it's very easy to publish that. Our project that I did, like that we were working on, was really difficult to publish because a lot of reviewers of this work said, you're not contributing anything new to science, so we're not going to publish this. Show us why this tool is so important or whatever. And so it took a very long time to get this published, even though loads of people were already using it when it was only a preprint. So I think in biology, it can be tricky sometimes, but deep learning is a real buzzword. And I think if you do something new with deep learning that nobody has done before, it's very easy to publish. And I've seen many papers that have used deep learning in a very, I would say, in a very simple kind of way that have been published in Nature Methods, where I think I don't want to sort of, those papers are still important and show novelty, but we could replicate a lot of the results that they had very easily on our platform. So it's not like sort of rocket science. A lot of it is just using an algorithm and using different type of data and showing that you can do A to B with deep learning, and then you can publish. It's quite, I mean, that is easy. It's harder to show that old algorithms can be used in a new way. I like, I guess like what we did was like basically building a new platform, and I think that was a big step forward. Yeah.
Okay, thank you.
Mm.
Well, I believe there is no questions anymore.
Cool. I hope I answered everything to satisfaction.
Yes, of course. Lucas, so I want to thank you very much, and to invite you again to come to Brazil and visit our country. It's it's a nice country. And thank you for your time, your you're disposable to talk to us.
Yeah, love it.
And I hope we can listen more about your research, because it's very interesting.
Thank you. Yeah, thanks again for the invitation. Always happy to do something like this. And yeah, maybe we can stay in touch and speak again in the future.
Okay. So I believe it's the end. We're coming to the end. I'd like to thank you, everybody who is watching to thank our audience, and say that this video will be in the channel of the Academic League of Histology, and in the future we'll subtitle in Portuguese this speech for those who don't understand English. And that's it. Lucas, thank you very much.
Thank you.
I hope we'll see you later.
Yeah,
again.
Sure. Goodbye.
Goodbye.
I don't know how to end this talk. Are you
I can end.
Okay. Cool. Bye.